So we're basically talking about bacteria. I think we're gonna cover gram-positives and leave gram-negatives for the next week. So what was I about to say? Yeah, there are certain things about each bacteria that I simply will not ask. Let's get it out of the way first. So for each species or genus, I will not ask about virulence factors. I will not ask about treatment. Now, with the virulence factors, I need to kind of correct myself a little immediately. There are 4 or 5 virulence factors that are on the addendum to the study guide. You may have seen it on the bright space. Those I can ask. I will make it abundantly clear as we go through the microorganisms, what you need to know, what you don't need to know. For every microorganism, as always, you need to know whether it is gram-positive or gram-negative. What is the transmission route? Okay? And whether or not there's vaccine. Obviously, you need to know the disease. So, to exemplify, let's start with Listeria. So, Listeria is gram-positive. We're going to talk about gram-positive bacteria now. And disease-wise, it can cause neonatal meningitis or meningoencephalitis in immunocompromised individuals, but it's one of the main causes of neonatal meningitis. In otherwise healthy adults, it causes gastroenteritis. Now, how severe this gastroenteritis can be? One of my former students here at Tri-C caught Listeria mid-semester, ended up in the hospital for 2 weeks, spent several days in ICU. Remember, it's a foodborne disease. Like, you can get it from eggs. And she was also pregnant at the moment. So she had miscarriage. Yes, because of Listeria. It's not a joke. Like, it is a severe disease. It is a reportable disease. Okay? Huge problem with Listeria in food industry. And this is kind of an cool story. You can imagine that if you work in a kitchen of a restaurant, you want something more practical than bleach for cleaning the surfaces. Well, because it's bleach, it's toxic. So there is now approved cleaning agent based on phages, viruses of bacteria. You basically spray the phage and it destroys Listeria. Leave it for 15 minutes. It's very specific for Listeria. It's not going to destroy viruses or something else like this. But for Listeria, it's great for Listeria control. Okay? And it's safe to add it to food. So even if the food is contaminated, you can add phage and it will obliterate Listeria within the food. Okay? Another cool gram-positive microorganism here is Mycobacterium leprae. We already learned about Mycobacterium tuberculosis. This is the relative, but it causes a different type of disease. So just like Mycobacterium tuberculosis, it reproduces in the macrophages. And when it infects macrophages, as the result of that infection and inflammation, the body forms granulomas surrounding infected phagocytic cells. On top of that, your body is trying to kill these infected macrophages using non-infected macrophages and CD8 T cells. That immune response results in the damage to the surrounding tissues and is responsible for lepromatous form of leprosy, which is more severe. Less severe form is called tuberculoid. So you see that gentleman here. Okay. In this case, bacteria are sitting in the granulomas. It is disfiguring, but at least the rest of the tissues remain intact, if we can say so. Again, in lepromatous leprosy, tissues quite literally are broken apart. Okay. At some point, it was a real scourge in many regions of the world. In the United States, leprosy was very common after, let's call it, acquisition of Hawaii, when Hawaii was protectorate but not the state yet. And it resulted in a pretty tragic page in the history of the United States, among many others. People who had leprosy or had a contact with someone who had leprosy were quarantined on the island of Molokai. But it is early 20th century, so no antibiotics existed. They were pretty much sentenced to a slow death, because even if you didn't have it, you would. Transmission is contact between people and rarely respiratory. And leprosy is zoonotic. It gets to people from animals, and the main reservoir are armadillos. So if you see one, don't try to grab it. Although it's really hard because they're really fast. I tried. Still, there are a lot of leprosy cases. You can see that even right now, we're talking about 200,000 people getting leprosy every year. So it's a pretty substantial problem. And the treatment is problematic because it's a Mycobacterium, so it reproduces slowly. So you have to maintain antibiotics for an extended period of time, which can be costly for patients and also have dramatic side effects. Now, another acid-fast kind of bacterium is Nocardia. So Nocardia, specifically Nocardia asteroides, is a classic opportunistic nosocomial infection. It's found in soil, which means it's found everywhere, and it can infect hospital patients who are in the hospital because of what? Why are people in the hospital? Because they're sick, and if they're sick, the immune system is suppressed. So that's the opportunistic part. So it can cause, in those patients, it can cause pneumonia, endocarditis, or encephalitis. So the infections of the lung, the heart, and the brain. From the perspective of microbiology, can you think of sort of a common feature of these three locations in human body? Those three locations are, from the perspective of microbiology.
Oh, sterile?
Exactly. Very good. They're sterile. And as any sterile environment, they're susceptible to establishing an infection. You will see this pattern as we go through other nosocomial infections, opportunistic infections. They prefer, if I can say this, sterile sites, because sites with resident microbiome, just competition is too tough. Okay? Now, this is the respiratory transmitted pathogen called Corynebacterium diphtheriae, which causes diphtheria. Essentially, one second, what just happened? Do you know? Okay. Effectively, bad pharyngitis. Bad is kind of an underestimate. Okay? So, during diphtheria infection, epithelial cells of your pharynx die, white blood cells die, and red blood cells die as well. And when all these dead cells start clumping together, they form a pseudomembrane. The pseudomembrane can grow so large that it can obstruct the respiratory passages. Diphtheria is a really serious disease with a significant mortality in people who are even treated but not vaccinated. Thankfully, it can be prevented. The disease can be prevented by vaccine. I try to put a little asterisk on the vaccine preventable conditions. It's a part of DTaP. Now, first of all, how does it kill the cells? This is the first virulence factor, which mechanism I want you to know. So, do you need to know which exactly elongation factor this toxin inhibits? No. This. It inhibits translation. Cells die. Okay? Diphtheria is one of the most contagious infections. I'm going to give you an example of, the vaccines, how effective they are. When I was in middle school, which was late 80s, okay, Soviet Union was already kind of collapsing-ish, okay, maybe it was 89, maybe it was 90, and some people started to reject vaccinations, and pretty much same year, there was a pretty large outbreak of diphtheria, several outbreaks across the Soviet Union. And it was stopped by vaccinating kids. And I can tell you, I remember how the process was. So we were sitting in the classroom during the lesson, okay? A school nurse would come in, pull us out one by one, walk us to the station, give us a vaccine, and we would go back to class. You may have noticed that I did not mention a single time any form of consent. Our parents were notified after. Nobody asked the permission. It was like, by the way, your son got vaccinated today. That's it. Stopped outbreak. Okay? Now these are, I generally say gram-positives are boring. So these two gram-positives, actinomyces and Gardnerella, I think boring. So actinomyces are present in the human microbiome, specifically in the mouth, and are implicated in gingivitis, the inflammation of the gums. They also, in people who are immunocompromised, they can get into the heart, because remember, every time you brush your teeth, you have bacteremia, so they may potentially infect heart valves, causing endocarditis, lungs, causing respiratory distress. They also can infect connective tissue, causing the condition known as a lumpy jaw. Okay? Gardnerella is an STD. Very interesting STD. It affects women much more than men. It's a little puny pathogen that doesn't do well in the competition, but when a woman starts taking antibiotics, this therapy suppresses the population of lactobacilli that form most of the vaginal microbiome, and Gardnerella raises its ugly head and causes vaginosis. Can be treated with antibiotics, so not a huge problem. Okay? And usually Gardnerella does not go farther than the vaginal mucosa. So it's not an STD in its classic sort of terms. Now, these three guys, Enterococci, Propionibacterium, and Mycococci, are all members of the human normal resident microbiome. As you can imagine, Enterococcus faecalis is the part of your GI microbiome, and when it is confined, to the limits of your intestines, it isn't bad. Does that make sense? Like, it's fine. It sits in the intestines. The problem starts when it escapes, and it can cause a variety of nosocomial infections, such as urinary tract infections, endocarditis, septicemia, and even pneumonia. The problem with Enterococcus is that it can be fairly antibiotic resistant because we take a lot of antibiotics that end up in our intestines. Then we have Propionibacterium acnes. These little guys. These bacteria are in hair follicles. When we enter puberty, our sebaceous glands that surround hair follicles start producing a fair amount of oil that contains short-chain fatty acids, and the sebum is used by Propionibacterium acnes as the source of nutrients. So when it overgrows, okay, it causes inflammation and acne. Okay, makes sense? A couple of comments on this. First, I don't know if you remember that many of you were not really conscious back then, I presume. The Deepwater Horizon oil platform, oil rig explosion, yeah? So we were in Louisiana at the time. It kind of, sort of, we were in the northern Louisiana, so it kind of affected just our recreational activities. We didn't go to the Gulf Shore for a year. But the scientists wanted to see how much it affected the tourism, how much it affected people going into the Gulf of Mexico. So how do you measure that? They did an absolutely ingenious data collection. They analyzed ocean water samples collected on the beaches because they regularly collected for quality over the period of time, and they were looking for the DNA of Propionibacterium acnes. Imagine how many people should have been using those beaches so that DNA was detectable, and they could match the popularity of those beaches to the levels of Propionibacterium acnes DNA in the water, which is kind of insane. Okay? The second comment, I don't remember if we talked about that. Popping the zit and smelling it, no? So, if I'm quoting, not my idea, folks. I can attribute it's Michael Schmidt, professor of microbiology from Medical University of South Carolina. Bacteria that ferment milk to produce Swiss cheese. I'm not talking about the sliced one, but really, really expensive and smelly. Belong to the same genus, Propionibacterium. If you pop a zit, spread it between your fingers and smell, it smells like Swiss cheese. That's the reason why socks and Swiss cheese share the same aromas. What, were you a fan of Swiss cheese? Not anymore? Wait till we get to sushi. Yeah. Huh? It's okay. I will ruin it first, and then I will restore your trust in sushi. Okay, so Mycococcus, lastly, pretty much benign, part of your skin microbiome. In extremely immunocompromised patients, can infect sterile sites. But I looked it up on PubMed, the database of manuscripts. There are tens maybe of cases, not a lot. And that brings us to Mycoplasma. I find Mycoplasma kind of cool because it is the smallest independently reproducing microorganism. Okay, you can grow mycoplasma on the agar plate, very special agar plate, but nevertheless. So, two mycoplasma pneumoniae, as you can imagine, causes pneumonia, walking pneumonia, and respiratory transmitted. Okay? Then we have Mycoplasma genitalium. I'm going to use this microorganism as an example of what happens in a sort of traditional urinary, sexually transmitted infection. There are some exceptions such as syphilis or HIV, but bacterial STIs behave generally the same. So first, the infection, like this is the female reproductive tract, but I will quickly explain male as well. So in females, stop number 1, vagina, so vaginitis. Then if bacteria spread to cervix, cervicitis. Then they can infect the epithelium of the uterus, endometriosis. And then they can get into fallopian tubes, so-called salpingitis. And out of fallopian tubes, they can technically get out into the pelvic cavity, causing pelvic inflammatory disease. So I want you to generally understand how it goes and what implications it may have for the reproductive function. Well, I'm not touching pelvic inflammatory disease. That's already bad enough, right? I have a couple of questions. First of all, the fertilization of a secondary oocyte. Where does it occur? In which part of the female reproductive system? Tubes, yeah. What happens to the fertilized ovum then? It moves to the uterus. What moves ovum? Which cellular structures on the epithelium of fallopian tubes? You know the answer, I guarantee you. Cilia. Cilia. I'm telling you, like, I'm not asking, you just need to dig in your knowledge, right? Now, when the ciliated epithelium is inflamed, inflammation begets repair, and as a result of repair, what type of tissue is formed? Start with S. Yes, and that scar tissue does not have cilia. So now these cells can't properly move the egg, but egg keeps developing. Cells keep dividing, form blastocyst. Blastocyst becomes morula while still in the fallopian tubes. What's the term for it? Ectopic pregnancy. Remember, there are some basic Greek and Latin roots you should know. Ecto, outside. Topos, location. Not in the proper location. Endometriosis can lead to the troubles with implantation of the blastocyst into the wall of the uterus. It's basically infertility. In males, since males share, since semen and urine share a part of the pathway, it starts with urethritis, and then it depends which turn the infection is going to take. If it's a turn into vas deferens, okay, then it's going to infect testes and result in infertility. If it keeps going through the urethra, it's going to cause cystitis. Mycoplasma is the cause of cystitis, mostly in men, as far as I know. In women, it can infect urethral orifice and urethra as well, just a bit of a more, farther movement. Actually, we're going to go a little more than 20 minutes. Bifidobacterial lactobacilli. These are, quote-unquote, certified probiotics. Okay? All of them, actually. But Bifidobacterium lactobacillus are probiotics in particular. there's a lot of studies that show that simple consumption of those microorganisms can help alleviate symptoms or get rid of certain diseases like symptoms. It's been shown multiple times. People with different types of inflammatory bowel disease, such as ulcerative colitis, Crohn's, they benefit from probiotics. Okay. Another method that kind of, may sound crazy, but actually isn't. Women sometimes experience vaginal dysbiosis, usually as a result of antibiotic therapy. The treatment that actually works is rinsing vaginal microbiome with yogurt or buttermilk. What? There is nothing wrong with that. It contains microorganisms, okay? Obviously, I don't think it's recommended to drink it afterwards, but yeah. So, what is crazy actually? Jokes aside, hear me out. What is crazy? How much bacteria do you think you carry right now in your guts? Well, give me a mass. No, mass, like in mass units. Huh? Minimum 2 to 3 pounds. Just bacteria, huh?
Bacteria.
Yes. Your fecal matter is about 30% bacteria. Dead, mostly. Okay? So there are a lot of them, like seriously a lot. I don't think it's trillions. It's tens or hundreds of trillions of bacterial cells. Now, when you take a supplement, it contains maybe 1 billion colony-forming units. It's a drop in the bucket. And yet it works. I'm dead serious. Nobody understands how this drop in the bucket can turn things around. But it does. Okay? Now, why don't we mention any gram-negative probiotics? Not enough studies. Probably there are. So we're going to consider only gram-positive ones. Gram-negative microorganisms are much more fun to talk about. So first, we're going to talk about non-hemorrhagic, what am I writing? Hemorrhagic fevers. Non-hemorrhagic fever means that the main symptom of the infection is severe fever. All these guys share pretty much same set of symptoms. So we're going to start with 2 species of Bordetella, Henselae and Quintana. So Bordetella henselae is a zoonotic infection that's transmitted by arthropods such as fleas and ticks, and it's the causative agent of cat scratch fever, the disease made famous by the song by Ted Nugent. Can be very severe, can be relatively mild. The symptoms basically high fever, myalgia, arthralgia, headache, a garden variety. Nothing super special. Okay? These diseases, don't take them lightly. They can be lethal if untreated. Bordetella quintana, that is transmitted by lice, also has the name of the disease trench fever. When do you think this microorganism was first discovered and described, if it's a trench fever? In trenches. In 20th century. When did we have a proper trench war? WW1, yes. Lice were abundant, so it was existed somewhere in nature, but a lot of people, poor hygienic conditions, a lot of lice made it possible to spread among humans. And then there's a Brucella abortus. Now, Brucella abortus causes a condition known as brucellosis. It got so many names: Maltese fever, Banks disease. undulating fever. So why undulating, first of all? You see the temperature profile? Up and down, up and down. It's not much fun in people, but it is a terrible disease in cattle. When I was writing the slides and I was searching for the image for brucellosis in cattle, I decided not to put any. It is very graphic. What happens to a fetus, because it's a spontaneous abortion, what happens to fetus is really very unpleasant to see. Okay? I know, everybody starts Googling this immediately. Yes, I know. Now, is it common in the States? Not really. Is it common in the middle to low income countries? Yes, I had a student who was a member of Peace Corps, and she was in South America. She got brucellosis, got cured, it's antibiotics. Now, let's talk about something closer to home. First one is rickettsia. There are, a ton of different rickettsial species, and most of them cause human disease. So what is so special about 3 microorganisms that you see in this table? All of them are obligate intracellular parasites. They can't reproduce independently outside of the cell. Good? Actually, it allows them to evade your immune response. The most famous condition that is caused by rickettsia, at least in the United States, is a condition caused by rickettsia, known as Rocky Mountain spotted fever. It's characterized by the characteristic rash, and pretty severe symptoms, and mortality can reach up to 20%. Now, this, I just want to highlight, this is what I just highlighted. I don't know how to emphasize it even more. Those are typical symptoms of pretty much all of these infections. Other conditions include rickettsial pox and typhus. Okay? Does that make sense? Now, rickettsial pox, did I ask you about the show called House MD? Yeah? If you watched it. So there's one episode, spoiler alert by the way, there's one episode when they think that the patients have smallpox. And then they figure out that it's not smallpox, that it's rickettsial pox. So, mixing up symptoms of smallpox and rickettsial pox are absolutely inappropriate for a physician, because smallpox is characterized by a huge number of pustules all over the body. Rickettsial pox, usually much less of it. Okay? But for the sake of the show, I completely understand the idea behind that. Why do you think Rocky Mountain spotted fever? Oh yeah, transmission arthropods. Ticks, mites. Where do you think it's most common? Rocky Mountain spotted fever. Which states, huh? So again? This shows you how absurd sometimes scientists are when they name something. It's most common in Tennessee, Arkansas, and Kentucky. It was first characterized and isolated and characterized in Rocky Mountain National Lab in Helena, Montana. And they called it Rocky Mountain spotted fever. That's stupid. I agree with you. That's very stupid. But the name stuck. Then we have Coxiella burnetii, which causes Q fever. Also, zoonotic transmission can be transmitted from farm animals and domestic animals. Okay? So this is one of the reasons we pasteurize milk. Okay? And then Ehrlichia. It used to be called anaplasma, but for the reason unbeknownst to me, taxonomists decided to change the name. So there are two types of Ehrlichia, both infect white blood cells. They are transmitted by ticks. Symptoms are pretty much the same: fever, myalgia, arthralgia. I can tell you this: if you have dogs and your dogs like to hang out outside, this is what they can catch in the state of Ohio. It's actually quite common. Okay? So this is in the among Russian jokes. One of them is we refer to STDs as pleasure diseases. We say that all other diseases are because of the stress, and only STDs are due to pleasure. Chlamydia is one of them. Second most common sexually transmitted disease in the United States. What's number one? Anyone wants to venture a guess? No, chlamydia is number two. No, not herpes. No. This has to be a very sneaky disease that causes almost no symptoms. No. Trichomonas is pretty bad. No. HPV, human papillomavirus. Number one. Absolutely number one. Okay? So, chlamydia. Remember the description of what's going on during Mycoplasma genitalium infection. Similar stuff happens with the chlamydia infection. Okay, so vagina, then uterus, then fallopian tube, so on and so forth. This illustration, number 3, you see enlarged lymph nodes. This is the sign of lymphogranuloma venereum. So at this point, there is so much chlamydia being drained from the infected, in this case, urethra, probably, that those lymph nodes become really swollen. Okay, but that's not the end of the story. Obviously, it's sexually transmitted. But what if, first of all, which sex is more likely to not notice the symptoms of an STD? Females. Think about the anatomy. Male reproductive organs are literally sticking out. If something is dripping, we're going to notice. It's just way more visible. In females, it may not be noticed. Okay? So, as a result, women can be infected with chlamydia, not knowing about that. And imagine at this point, woman is pregnant and about to give birth. So, a child coming through the birth canal, finally, coming through the birth canal can easily acquire chlamydia. And chlamydia doesn't really care because it infects epithelium. And a very sensitive epithelium, skin is not really infectable, it's super dry. But really infectable epithelium is the eyes. So it can cause trachoma. Infection of basically it's bacterial conjunctivitis. If it's untreated, it leads to blindness. Okay? It's treatable with usually topical antibiotics, usually tetracycline ointment or tetracycline eye drops. But in low to middle income countries, access to antibiotics may be complicated. So one of my very former colleagues back in Russia, she used to work on the project, the vaccine against trachoma. And I actually made it. It was working just didn't have a market for it. But she said, to give an idea, trachoma is extremely infectious. You know, like if you have a child with pink eye is different, it's not, obviously. But even if you have a child with pink eye, better not allow them to be around other kids because that will be transmitted. Trachoma is even more infectious. According to her, when they were working, biosafety, hoods and everything, every person in the project got trachoma at some point. All precautions, safety measures, everything, super infectious. Okay. Let's do Bordetella and Neisseria because these guys are fun to talk about. Since we are riding the train of sexually transmitted infections, let's chat about Neisseria gonorrhea. Causes gonorrhea. Symptoms are similar. Basically, when you look at what I call classic STDs, gonorrhea, chlamydia, Mycoplasma genitalium, Trichomonas vaginalis, the difference will be the color, the smell of the exudate in males or females, but the whole progression basically is the same. So, interestingly enough, just like chlamydia, Neisseria gonorrhea can cause conjunctivitis in newborns if a woman carries gonorrhea during birth. Okay? Again, it's treatable. So this image shows you the child with the condition, sometimes referred to ophthalmia neonatorum, but it can be used for chlamydia infection as well. Okay? Again, treatable. However, I wonder what made it. You're familiar with antibiotics, especially fluoroquinolones, and the most famous of them is ciprofloxacin. So more and more strains of gonorrhea turn out to be resistant to Cipro. Now, what's going to come next is going to be our speculations. But I see a pretty good reason behind it. So imagine that you was diagnosed with gonorrhea. Be honest. And obviously, you had unprotected sex, now you have gonorrhea. Would you be embarrassed? Yeah, a little, you know? I mean, you're not going to call your friends and cancel your dinner saying, sorry, folks, I have gonorrhea. If you have flu, fine, right? Sorry, I have flu, yeah. Sorry, I can't come, you know? So, a bit embarrassing, okay? But you know, or you look it up and it says, you know, you can treat it with Cipro. So you may have a friend who's a physician, and you'll ring them up, say, hey, can you do a prescription? And they can, and you pick it up, and you start taking it, and you don't complete the entire course. So my bet is on people who just go for this treatment, don't complete the entire course, and foster the resistance. Okay? Does that make sense? I'm not blaming those folks. I'm just saying that that's something to think about. One of the nasty consequences of untreated, sort of chronic gonorrhea, it loves traveling to joints and to cause arthritis. It's a very common long-term consequence of untreated gonorrhea infection. Okay? Now, same genus, different infection. Neisseria meningitidis, also known as meningococcus, causes meningitis. There are three main causes of community-acquired. I'm going to put the word community here. It's Streptococcus pneumoniae, Neisseria meningitidis, and Haemophilus influenza B. This is, Neisseria meningitidis is probably the worst. there was a case, if my memory serves me well, when an unsuspecting lab tech took out agar plates from the incubator, not knowing that it's Neisseria that grows on them, and opened them. Enough bacteria aerosolized from the plates to infect him. He died in 48 hours. Died. Because it went straight through the nose, into the brain, caused meningitis. It developed super fast. It is not a joke. Okay? Thankfully, there's a vaccine. Okay? And I actually encourage everybody, when you visit your PCP next time, insist on checking the vaccine schedule, because we all often think, oh, we're adults, we don't need it. Yeah, maybe we do. Okay? So it's a good thing to update it. Treatable, if everybody would get vaccinated, we could probably eliminate it, because humans are the reservoir. Now imagine everybody's vaccinated, bacteria don't have any place to reproduce, and eventually dies out. That would be awesome. And then Bordetella. Bordetella pertussis, another vaccine-preventable infection. It's a respiratory infection, also in humans. We also can eliminate it. It causes whooping cough. Okay? Also known as pertussis. So this microorganism has 3 stages of disease. First is catarrhal. That is actually the peak of infection. Catarrhal pretty much means with a cough. So people cough a little bit, runny nose, looks like common cold. And then the condition deteriorates when they enter the paroxysmal stage. Your trachea is covered by pseudostratified ciliated epithelium, which moves mucus upwards towards the larynx, so-called mucociliary elevator. We talked about it in Unit 4. The toxin that's produced by pertussis destroys that epithelium and specifically destroys the cilia. So mucus stays with all the crap that it absorbed. And the result of it, irritation of your trachea and a cough. I don't have the ability to click on the link, but in the actual notes, if you click on that link, it'll take you to the YouTube video of a guy, adult, coughing. Yes. You hear that? So the bouts of cough can be so violent that people would throw up and the blood vessels in their eyes will pop. It's called bloodshot eyes. Okay? This year, I didn't check the latest updates, but this year, in the state of Louisiana, for example, already 2 pediatric deaths. We're talking, unvaccinated kids. Dude, it's a Bible Belt. We're talking unvaccinated kids, and we're talking little kids. So they were definitely under 1 year of age. Because DTaP isn't given immediately, so kids may be unvaccinated, not because of the failure of the parents, but because they're just not eligible. Okay? Now, an interesting thing about pertussis, the original vaccine was done in the way that you just take Bordetella culture and just inactivate it, kill it. So whole cell vaccine was given. It worked great. Side effects were horrible. Not in the way. Basically, you receive DTP and you floored for 2-3 days. Kids like headaches, sometimes seizures, super high fever, bad reaction. Nobody died, but bad reaction. They figured out it was because of the fragments of LPS, lipopolysaccharide, from gram-negative Bordetella. They managed to remove it. This is why the vaccine now is called DTaP. Do you have that phrase? No, I don't. Which stands for diphtheria, tetanus, and acellular pertussis. Unfortunately, with a decrease in side effects, the efficacy also went down. So now it is considered, one shot is considered to be 80 to 85% effective, so we take multiple shots, we take boosters, okay? Does that make sense? So I'm trying to make, I'm trying to make a case for the vaccines protecting other people. You know, when somebody says, oh, I'm not taking a vaccine, I'm going to get sick. Well, you can transmit it to a person who can't get a vaccine for a reason, such as 3-month-old child, and the outcome will be very different for that person, okay? And also, how great would it be if you could just just vaccinate everybody, right? That would be awesome, okay? So we're going to continue the conversation about various gram-negative microorganisms, which brings us to the group of bacteria called Enterobacteriaceae. And you also can see that there's a term commensal. These are bacteria that can be found in human gut in a different abundance. Okay? First of all, let's take a look. Remember, I told you need to know if it's gram-positive or gram-negative, the transmission route, and the disease. Let's take a look at the transmission route. All of them can be transmitted in the hospital settings. Many of them can be transmitted by autoinoculation because they are found in the gut. Okay, cool. Second, these bacteria, when they are in the gut, they're pretty much benign. They don't do much in there. But when they get into sort of a wrong place, they can cause a bunch of opportunistic infections. I'm going to highlight. So pneumonia, lungs are sterile, no competition, can cause pneumonia. Septicemia, blood is, or bacteremia. Blood is sterile, no competition, there you go. UTIs. Basically, again, think of those bacteria as the ones that are weak and can only establish themselves well enough to cause any damage in locations which are effectively sterile, like your urinary tract, like your lungs, or like your connective tissue. Okay? Now, we do not have, I told you, we don't need to remember the virulence factors, but I want to highlight something kind of important here. Where's the... Here. We finished, last week we discussed the gram-negative bacilli lab, and one of the actors in that lab was gram-negative Proteus vulgaris, similar to Proteus mirabilis. They're pretty much the same. So, and you remember that microorganism was able to break down urea. Okay? So it turns out, if this microorganism ends up in your kidneys as a result of untreated urinary tract infection, it's going to have plenty of urea around, and it will break it down all right, and the release of carbon dioxide and ammonia as the result of urea breakdown will very much speed up the accumulation of kidney stones. Now, these bacteria come from the gut. So a part of one of the opportunities, like for the transfer of those microorganisms from the gut into the urinary tract, is poor hygiene. Does that make sense? Well, first of all, who are more prone to urinary tract infections, men or women? Women because, and I insist, I am quoting another scientist, literal quotation from Dr. Michael Schmidt, the professor at Medical University of South Carolina, because women have intrinsic problem with plumbing. The anal orifice and the urethral orifice are too close. That's anatomy. In men, the distance is farther. But here's another thing. What do you think if a person, let's say a person wants to pee, but they decide to hold the pee for as long as they can? Does it increase the risk of urinary tract infection or not? It does, because when you pee, you flush the stuff out. And there are probably few groups of people that prefer to hold their pee. One of them is truck drivers. And actually, bacterial infections of the kidneys and urinary tract infections is sort of a professional risk for those folks because they sort of facilitate it. Okay, also, as you can imagine, maintaining personal hygiene, being a truck driver becomes a little challenging. Okay? Now, the sort of most famous Enterobacteriaceae is, of course, E. coli. Now, when you start looking at the table that I created here, okay, you may notice that the transmission is nosocomial autoinoculation, or it can be water or foodborne, meaning that when you drink contaminated water or eat contaminated food, you can get this infection. Now, the common feature of pretty much all species, all strains of E. coli is that they can cause diarrhea. Okay? Do you need to memorize all those individual strains? Of course not. But there is one strain that I want you to know and be aware of. It is enterohemorrhagic E. coli, or EHEC, also known as O157H7. Please do not memorize that word soup. Okay? So enterohemorrhagic means that there is bleeding in the gut. Does that make sense? The bleeding in the gut is called dysentery. Now, if you think about the gut and the barrier between the blood and the gut lumen, if there is a blood, that barrier has been breached, which means it actually can go both ways. Just like blood can enter the gut content, namely that EHEC, can go in the blood. Turns out that enterohemorrhagic E. coli produces toxins called Shiga toxins, which result in the cell death. And they can kill red blood cells. Killing red blood cells will cause hemolysis. Release of heme from red blood cells will lead to acute kidney failure. And when kidneys are failing, they can't excrete urea properly, so urea starts accumulating in the blood, causing hemolytic, that's breakdown of red blood cells, uremic syndrome. Presence of urea, which is alkaline, increases blood pH, sends the person into alkalosis with all the consequences of it. Okay? Now, where do you get this relatively nasty bug? Food, for the most part. So those, you may have heard the outbreaks when in the news they say there's an outbreak of E. coli associated with romaine salad or bean sprouts or some stuff like that. It actually also can come from meat. And speaking of meat, microbiologically, which food is safer? Medium rare steak or a medium rare burger? Why? Huh? Yeah, because burger is chopped up. Because in burger you mix the meat. In a steak, the meat, the muscle, technically speaking, is sterile. I mean, I'm not talking about parasites, that's a separate story, but not likely. There shouldn't be a lot of pathogens inside the meat. So when you sear the steak, you kill whatever's on the surface, and you're pretty safe. When we get to the burger, take your chances. Okay? Make sense? I tried once the uncooked ground meat, the national dish. Here I am still alive. Okay. Now, where does it come from? E. coli is abundant, not only in humans, but in animals as well. And if you think about greens that you consume, they grow usually in the open field, and animals will pee and poop around, you know. So wash your greens, even if they are pre-washed. It's an official recommendation of the Food and Drug Administration and CDC. Okay? All this pre-washed stuff is riddled with fecal microorganisms. Now, we're kind of building up the suspense. So now Shigella and Salmonella. Shigella is the human pathogen, which is transmitted by a fecal-oral route and also has Shiga toxins, which inhibit translation and cause cell death, just like E. coli has. Okay? Very similar symptoms to enterohemorrhagic E. coli. Diarrhea, then dysentery. So this image number 1 shows you red and white blood cells in the stool. Okay, now, unlike Shigella, another enteric microorganism, Salmonella, comes from animals through poultry and eggs, and dairy to the less extent. But now, why poultry, surprisingly? Biologically, birds, including chickens, are pretty much feathered reptiles. Not entirely, there are other distinctions, but it's basically a reptile. All reptile species, pretty much, carry Salmonella in their guts asymptomatically. So if you have a pet lizard or pet snake, they do have Salmonella. We have a snake at Lakeland, the department snake, and we have department lizard as well. We tested lizard poop, Salmonella was there, just on the average day. Okay. So mild salmonellosis manifests as enteritis. I experienced it once when I was 9. I'm good. I don't want it anymore. I thought that I will be turned inside out during that process. More severe forms, typhoid and paratyphoid fever. This is a systemic infection. It is a serious cause of mortality in countries that lack or have insufficient water purification capacities. So much so that if you travel to the countries where you may experience shortage of properly treated water, there is a vaccine against typhoid fever that you can get in the United States. It's only for travelers, but you absolutely can request that vaccine from your healthcare provider. Okay? And it's really effective. So why? Maybe, we just should tough it out. So when people recover from salmonellosis, they still carry bacteria in their guts for up to 4 weeks. Now imagine you come back from that trip and you start spreading it around, which you can. So that's a good reason. Moreover, Salmonella can colonize human gut asymptomatically. It is rare. I don't know if you heard of the story about Typhoid Mary. She was a cook. She was from Ireland. She was a cook in New York in early 20th century. I think it was like 1920s, 30s maybe. So in the houses where she was cooking, people went down with salmonellosis. They figured that it comes from that Mary and told her, please don't be a cook. That went swimmingly because she realized that she can't earn enough money by not being a cook, and she came back to, being a cook. The outbreak started again. They grabbed her and quarantined her on Plum Island. It's a bacterial facility, infectious control facility. But in fact, she was imprisoned. It wasn't a prison like you have a horrible cell. No, she had nice accommodations, but she couldn't go anywhere. She couldn't work anywhere. And eventually, much later, U.S. government paid her family money to settle because they sued because it was imprisonment without a trial. Okay, that kind of stuff. Now, this is one of my favorite microorganisms. It's Vibrio. So there are two species I'm going to be focusing on, Vibrio vulnificus and Vibrio cholera. So this is Vibrio vulnificus. All Vibrio species don't need humans. They can reproduce in shellfish. They can reproduce even in water. And they prefer specifically water that is kind of a little salty, so brackish, basically the interface between the fresh water, like a river, and the salt water, like the ocean. Okay, they'll like it. So Vibrio vulnificus is one of several flesh-eating bacteria. They cause wound infections, which cause necrotizing fasciitis and very bad degradation of the tissues. So traditionally, these infections affect, interestingly enough, people who surf because it's ashore and they get scraped and bruised and stuff like that. But a few years ago, there was a hurricane that affected the Gulf side of Florida. It really hit Fort Myers bad. So they had local flooding, and effectively the fresh water and the water from the Gulf mixed. And there were several cases of Vibrio vulnificus in Fort Myers and surrounding areas because people were just they were wounded and they walked around in that water, whatever. But the most famous one is Vibrio cholera. So this guy causes watery diarrhea. You can see I use the adjective profuse. Bouts of diarrhea every 20 minutes. People lose insane amount of fluids, which results in acidosis, as well as hypovolemic shock, and they die. So first of all, let's talk about how it causes. This is one of the virulence factors that I want you to know. So the toxin that's found in cholera stimulates the enzyme called adenylate or adenylyl cyclase. You may have heard about that enzyme in your A&P. This enzyme produces cyclic AMP signaling molecule, which signals to open channels facilitating the secretion of ions into your gut. Water follows ions. So water will leave too. You have watery diarrhea. Okay? Now, if somebody gets cholera, what can you do? This. The most important component of the cholera treatment is fluid supplementation. It's like it's really hard to drink that much electrolytes. Okay? So these folks have the IV form. Okay? Now, where does it come from? It comes from water. I don't remember which, like, it was taken, this image was taken from some paper about cholera to illustrate that sometimes water sources aren't living up to your expectations. And the first question that you should ask is, can we boil the water? You absolutely can, and it will be clean. The only problem is that you have to cool it down and you have to spend energy, which for families who are poor may be an issue. They may not have enough resources to boil all the drinking water. So how can you control the infection? Sort of cheaply, but fairly effectively. I'm going to tell you how, and you will try to hypothesize how this works. It would be wonderful if you could filter the water, right? But to filter all the bacteria out, you have to have a pretty thorough filter. It turns out, though, that if you take cotton cloth, the type of fabric that is used in India to make sari, and fold it 4 times, 4 layers, and you filter your drinking water through 4 layers of fabric, which isn't a huge deal, right? That dramatically reduces the risk of getting an infection. Obviously, cloth isn't that thorough of a filter to remove bacteria out of the water. So my question for you, how does that work? Huh? No. Take a look at the transmission routes for cholera. Read it out loud. Contaminated water. What's next? What is shellfish? Yeah, crustacean, yeah.
Fish with shells.
Pretty much. Non-kosher, though. Can you filter out crustaceans? Yeah. So crustaceans don't have to be big. We're not talking about shrimp, swimming around with cholera. We're talking about small crustaceans, the ones that, remember when we did this pond water thing? Microscopy? Some of the moving stuff there, that was the crustaceans. Turns out 4 layers of cotton is enough to filter them out. And they carry the bulk of cholera. So you get most of it out. And since median infectious dose is pretty high, you're good. Now, another thing that we should be aware of, that there was a case of asymptomatic carriage of cholera in the guts. It was in Haiti when they got hit with an earthquake quite a few years ago. The country of Nepal sent a bunch of troops to help with, recovery mission. And shortly after, since water treatment facilities were destroyed, there was an outbreak of cholera in Haiti. And they traced it to those Nepalese folks who, turns out, were carrying cholera in their guts, showing no symptoms whatsoever. So now it's established. However, there's something we should be thankful to cholera for. It's a birth of the science known as epidemiology. I believe it was 19th century, fairly early, in London, in the area where mostly like blue-collar workers, poor people, lived. There was an outbreak of cholera, and the physician who was treating those patients, named John Snow, apparently he did know something. John Snow traced all those cases to a single water pump where people collected water for drinking. And basically he laid out how do you determine the source? How determine the transmission? So he managed to question and trace the source. What's interesting though, so people who sampled water from other pumps were fine. What's interesting though, the people who got sick were only women and children. Men did not get cholera. Do you want to venture a guess why?
Maybe they drink outside the home.
Well, can you be a little bit more specific?
They're drinking water.
They're not drinking water, beer or whiskey. That kind of shows you that water is dangerous. Now, cholera, by the way, cholera is still a huge problem. So this map shows you the outbreaks of cholera. It's not like it constantly is present, but there are outbreaks associated with usually waterborne transmission. And we are finally at the apex in terms of the sort of how interesting it is. So we're going to talk about the genus Yersinia. Most of the members of genus Yersinia, and I use one as an example, Yersinia enterocolytica, they cause some kind of gastrointestinal disorder. They may cause sepsis sometimes, but for the most part, it's diarrhea and vomiting. And it can last for several months, usually contracted from contaminated food. Contaminated by whom? Semi-feral dogs and cats. Yersinia enterocolytica is another microorganism found in birds and in reptiles. And if they run around and they chase something and kill it and then bring it back and passes through the gut, so yeah, possible. But it's not the most famous species of Yersinia. This is. It's Yersinia pestis. Okay? It's a causative agent of plague. So, how is this disease transmitted? The reservoir for the plague is rodents. There are several sites around the globe that still have plenty of plague cases. In the United States, cases of plague occur west of Rocky Mountains. As you can see, there used to be not a lot of cases up north, like in Montana or Idaho. These numbers keep climbing because of the climate change, the warming. Infections move up north. Okay? So, fleas that feed on rodents can be directly transferred to humans, or they can jump on your dog or cat and then jump on you. So which rodents house plague in the United States? Prairie dogs. Chances that you will catch a prairie dog are pretty slim. Chances that your dog will chase one are much higher. So my point is that initially you, human, have to be bitten by a flea. And there's a very interesting mechanism that facilitates the transmission, and this mechanism comes from the bacteria itself. So imagine there's a flea, it sucks the blood of a rodent. That blood contains plague. Now, it may take some time for that flea to digest all of it. In order to prevent the digestion, bacteria seal shut the digestive system of a flea so it can't poop bacteria out. Then the flea, when it ends on a human, tries to take another blood meal from a human. Now remember, at this point, flea is full of a rodent blood, so it has to dump it. The only way it can dump it is vomiting blood out. So when it takes a blood meal, it empties its gut, it vomits the blood from the rodent into human circulation. And that's how bacteria get there. all because it blocks the digestive system. Once the bacteria are in the blood, they travel to the closest lymph node and establish the infection of the lymph node. This is called bubo. This is why bubonic plague. In case of bubonic plague, if untreated, mortality can reach up to 70%. And if bacteria make its way to the lungs, it can result in the pneumonic plague, which is pretty much as anything pneumonic is awful, with a much greater mortality rate. If Yersinia pestis starts to reproduce in the blood, effectively causing septicemia, shown here. This is the necrosis of peripheral tissues in septicemic patient. You see black toes. They necrose, they gone for good, okay? The mortality rate of untreated septicemic plague is 100%. Untreated. Plague can be treated with antibiotics relatively easily, okay? Now, couple of sort of trivia bits. You probably have heard about the plague pandemic in Europe in early 14th century. It wiped out about half of European population. And it also was the defining factor that created capitalism. A lot of people who were skilled workers died. A lot of people who needed their services, like rich folks, didn't die because they could afford living alone. So suddenly the job market tilted in favor of those skilled workers like carpenters and blacksmiths and you name it, masons, and they started charging more money. And that effectively created a middle class, which was, it is 100% true, plague pandemic was the foundation for European capitalism. Okay, for industrial revolution in Europe, eventually. Another interesting thing you didn't notice, but all plague cases in the United States or in the West, there's one little case right here in the city of Chicago. City, not like prairies of Nevada or something. How did that happen? There was an associate professor, I believe it was University of Chicago, but there's two, University of Illinois, University of Chicago, one of those. And he studied, guess what? Plague. And he was developing an attenuated vaccine. So one night he showed up at the ER saying that he's not feeling well. He was evaluated and was told, nah, it's probably a virus. So next evening, he was brought back into the same ER by the ambulance. He was delusional, super high fever, bad shape. He was placed in isolation. His blood was, sent for cultures. He died 24 hours after the admission. The blood came 24 hours after he died. Turns out he had plague. He was supposed to work with attenuated strain. What happened? There's an article in Morbidity Mortality Weekly Reports that was published on that case. So it turns out that dude had a genetic condition called hemochromatocytoma. It's a condition when people have abnormally high levels of iron in the red blood cells. And Yersinia pestis reproduces absolutely crazy when there is iron. So in a regular folk, it would probably be some mild bacteremia, treatable, In that guy, it was such an explosive growth that he became septicemic in a matter of a couple of days. Okay? It was real case, real investigation. Icing on the cake. According to him, what he managed to tell people before the physician before he died, at that time when he worked and got inoculated, he didn't wear gloves. Which is, yeah, classic me. I wouldn't wear gloves either, especially if nobody's in the lab. Okay. Now, yeah, this one, Francisella. It's not common, but it happens in the United States. It's also known as rabbit fever, mostly found in rabbits. People get exposed either via the skin. So this is cutaneous tularemia. Or they can inhale it by respiratory route. And there were interesting cases when, remember, rabbits are reservoirs, right? People would be mowing the lawn, and there would be a rabbit, and like ride mowers, and they would run over rabbit, vaporize it, inhale whatever gets out. The infectious dose is very small. Okay, pretty bad. Not frequent. So there is a vaccine, but it is not recommended for general population. If you work with tularemia, usually get the vaccine, just in case. Okay. Now this is a pretty like variable bunch. I'm missing the word. Pseudomonas aeruginosa. You will definitely encounter that microorganism one way or another in your clinical practice if you're going to be working directly with patients. It is an environmental microorganism that is transmitted mostly in the nosocomial settings in the hospital environment. It survives on pretty much anything. It has notorious drug resistance, like insanely drug resistant. And it can cause a bunch of pretty cute diseases, such as, and I'm not making these names up, green nail syndrome or hot tub rash. Oh, indeed. Yeah, if you think about hot tub is just a bath water from other people. Okay. Now, the most common population, the population that is affected the most, patients with cystic fibrosis. So, for a decent period of time, these patients with cystic fibrosis received prophylactic doses of antibiotics to prevent the infections, which wasn't the greatest idea because prophylactic doses, low doses of antibiotics, they foster antibiotic resistance. So now there are totally drug-resistant strains of Pseudomonas that refractory to regular doses of antibiotics. It's a pain in the ass to treat them. And there were cases when people were denied lung transplants because they were positive for totally drug-resistant Pseudomonas. And you can't just remove a lung, give a new lung, and have it spread because they would have to be immunosuppressed, right? And one of the cases, to be fair, to be completely correct, it wasn't Pseudomonas, but it was kind of relative, Burkhalderia mallei, a girl from Columbus. Could not get a lung transplant because she had totally drug-resistant bacteria. So parents and the girl asked scientists who study bacterial viruses, bacteriophages, to make a concoction to treat Burkhalderia. And they did. And they nebulized those phages. And the girl inhaled them, and in a couple of weeks she was clear. That's phage therapy is a very powerful thing. Legionella pneumophila. if you like going to Cedar Point in the summer, you have a pretty decent chance of exposure. Legionella normally infects amoeba, and amoeba live in the water. And if there is an open water that can be aerosolized, like a cooling tower, these droplets can contain amoeba with Legionella inside them. If those amoeba get inhaled, Legionella gets out, looks around, sees, well, the only amoeba-like cell that I see around is an alveolar macrophage, gets in there, and causes pneumonia. Okay, also known as Legionnaire's disease. It's called Legionnaire's disease because it was discovered first cases were identified during the American Legion meeting. Quite a few people died. What's interesting about it, it is not non-communicable. Okay? Now you may see Pontiac fever. Pontiac fever was a milder outbreak of Legionella in the town of Pontiac. I think it's Michigan. And then Haemophilus influenza. It has nothing to do with influenza. It's not flu. It's a respiratory transmitted pathogen that can cause upper respiratory tract diseases, but the most important syndrome that I want you to know, meningitis. It completes the unholy trifecta of Streptococcus pneumonia, Neisseria meningitidis, and this. Three most common causes of community-acquired meningitis. If anyone has a school-age kid or kids, and they go to a public school in the state of Ohio, they must be vaccinated against all three, or they won't be allowed into school. Okay, so all three are vaccine preventable. Sounds good? H. pylori. The only reason why it occupies the entire slide is because this illustration on the right is absolutely awesome. So H. pylori is microaerophilic and capnophilic. So it needs very specific levels of oxygen and CO2. And what's interesting, it doesn't like the acidic environment of the gut that much. So when it is in the gut, it tries to escape by releasing the enzyme, on which name of which I don't remember. So basically that enzyme destroys mucus. And then bacteria infect the epithelium of your stomach. And there's a double whammy here. Bacterial infection causes inflammation, and the disrupted mucus cover also exposes that inflamed ulcer. Am I making sense? Long story short, people end up with what's called peptic ulcer. Okay? Folks that discovered it, Marshall and Warren, couldn't figure out how to prove definitively, because they found it in the guts, in the stomach of all patients with peptic ulcer. They wanted to see causation. And then I think it was Marshall. One of them was a scientist, one of them was a doctor. So the doctor just got the beaker of bacterial culture and drank it. Got classic pathology, replication, got treated with antibiotics, ulcer resolved, so all conditions were met. You can eradicate it basically here. So you see up more than 90% eradication efficiency. Now, prevalence. The country where I was born, Russia, yep. It's very common. Super common. Okay. Like one of my classmates actually died of peptic ulcer that got through the stomach wall. Okay. And these three guys. I'm going to start with the boring one at the bottom. All three of them are spirochetes, which means they are fairly thin and often hard to see using regular bright field microscopy, but the dark field microscopy works marvelously well because, you know, they start being shiny. Okay. So Leptospira interrogans. If you have dogs, cats, I'm not that much familiar with cat immunization practices, but if you have a dog, I hope you vaccinate your dog. And one of the, no? Yeah? One of the components of annual vaccine is leptospirosis vaccine. Okay? Because your dog can wander. I don't know if yours does. I don't know. But if your dog wanders around, it may chase other animals and get infected. And then Leptospira can be transmitted by fecal or urinary route, feces or urine, to people. Usually it's a fever, okay? But in some cases, it can lead to liver and kidney failure, okay? Make sense? So this is classic zoonosis. Another classically zoonotic infection is Borrelia. 10 years ago, I would tell you that Ohio is not a hotspot for Lyme disease, but now it is. When I say hotspot, it doesn't mean that, you know, you get out of the door and there are like 150 ticks waiting for you, but it became definitely way more frequent and kind of nears New England, which is a super hotspot. Okay? Transmitted by tick bite. And sometimes, but not always, presents itself with that bullseye rash. Now, if you notice it, it's probably Lyme. If you find a tick and there's no bullseye rash, it means nothing, because it's not always happening, this rash. Technically speaking, if you found a tick on yourself, what are you supposed to do with the tick? Just remove it. You got to remove it. What are you supposed to do with a removed tick afterwards? Huh? I mean, that's fun, but not as much fun as burning something bigger. Okay? You're supposed to freeze it and bring it to your healthcare provider. And they are supposed to send it to CDC. I believe now, healthcare providers, the only thing that they send to CDC are condolences. But technically, CDC is a reportable, CDC. Lyme disease is reportable. So CDC should say, okay, this tick came from Cleveland, Ohio. We need to check if it contains or doesn't contain a range of tick infections. They don't really do it. How many ticks are positive? Usually about 0.5% of tick population carry the disease. If they just beat you and didn't really engorge with blood, chances of transmission are really low. Okay? It's treatable. The longer you wait, the worse the outcome is going to be, because it can get into your joints, causing arthritis, and it can have severe neurological consequences, such as paralysis. Not the entire paralysis, but certain parts. Okay? And the really good disease, well, not good, but you get my drift. Treponema pallidum causes syphilis. Transmission entirely sexual. Well, there can be congenital, but let me clarify what I mean by entirely sexual. Syphilis cannot be contracted by kissing, hugging, sharing the plate. Just can't. There should be a transmission from one reproductive epithelium, genital epithelium, mucosa, to another genital mucosa. That could be done, obviously, through sex. There were reports of syphilis transmission by sharing a towel, and there was a report showing syphilis transmission. Don't ask me the context or circumstances. Some people have their own kinks. Sharing underwear. I agree. That's probably too much. Now, when it is transmitted, the disease starts with the primary syphilis. So primary syphilis. In primary syphilis, there are chancres on the genitalia. Okay? They are easier to spot in men than in women because of the anatomy of reproductive organs. If it is left untreated, it develops into secondary syphilis with symptoms such as mucocutaneous rash and low-grade fever. When I just started college, a good friend of mine was in medical school. We have slightly different system. College and medical school are not separated. It's one long 6-7 year program. And he had to write a term paper on secondary syphilis, so he got a book, like for physicians. And I was flipping through the pages of the book describing different presentations of secondary syphilis with colorful photographs, and mostly of the junk. Okay, and I had two questions for people, like rhetorical questions for people who were photographed there. First, if those people own a mirror, because even if you can sit down there, it's pretty obvious that this isn't right. And second question that I had, do you think that if it looks like that, and it was really bad, it's going to get better? It's kind of turning black. It's probably not going to heal on its own. Both primary and secondary syphilis are contagious. Tertiary is not. In tertiary syphilis, bacteria spreads to various sites, such as blood vessels and brain and connective tissues, can cause meningitis, can cause aortic malformations like aneurysms. This is what's called a gamma, an infection of a connective tissue. I didn't put any classic old end of 19th, early 20th century photographs of the tertiary syphilis. I know that you're going to Google it. You go ahead. It's fascinating. For instance, human nose, that part, the apex and the part of the septum, is cartilage and dense connective tissue. Treponema pallidum will eat it. Yes. That's pretty disturbing. Now, the thing is, back then, there was no treatment. So imagine, somebody gets infected. They know exactly what they're going to go through. All steps until they die. Okay? I often say that, syphilis was a gift. When European colonizers invaded Americas, they brought their own gifts, namely measles and smallpox, which killed more Native Americans than any weapon together, combined, times and times more. Syphilis, most likely, was given back by Native Americans to Europeans. So it's not exactly a fair exchange, but now we're going to switch to fungi. Fungi are not terribly interesting, seriously. They're okay. You will see why I say that they're not terribly interesting. There aren't a lot of fungal infections around. Classification-wise, just a reminder, molds, multicellular. Yeasts, unicellular. Molds form hyphae, the long threads that can be septate, meaning there are septa, separations between the cells, or non-septate, where cells fuse, so no membrane, no cell wall, right? And role-wise, hyphae can be vegetative, can be reproductive, vegetative, get food, reproductive, reproduce. Now, what about reproductive strategies? Molds can reproduce by fragmenting the hyphae. Yeasts can reproduce by budding. So here you can see a bunch of new yeast cells budding off of each other. So basically, kind of binary fusion, but fancier than in bacteria. Each cell is an individual organism, but they can remain stuck together. So this is why we call it a pseudo-hyphae sometimes. We're going to talk about spores in a minute. What is dimorphic fungus? Dimorphic fungus is the one that exists as a mold in the environment and yeast in the human body. Now, when I say environment, I mean like the parks, the forests, okay? So usually the temperature doesn't get higher than like 70s. The human body is a 90 something though. So most likely the change in temperature is the switch that turns molds into yeasts. Now, what about spores? I'm not going to be focusing as much on the structural features and more on the functional features. Spores come in different flavors, but the most important difference is that there are asexual and sexual spores. So asexual spores, illustrated by this sporangio, spores that come from the sac called sporangium, and these conidiospores, they basically clone a fungus. Does it make sense to you? Do you understand what I'm saying? Like, they clone it. It's a copy of the original. But when a fungus becomes stressed, it starts producing sexual spores, which work just like, male and female gametes. So sexual spores, male and female spores, fuse. They are haploid. So when two haploid spores fuse, they form a diploid organism. And that diploid organism has an increased genetic variation. So from the reproduction standpoint, they aren't terribly important. But from the evolution, adaptation, survival standpoint, they are. So they're not a main route, but they're formed under stress for that particular goal. Okay? Now let's talk about some fungal diseases, shall we?